Welcome to the deep dive. Today, we're exploring something called OakVar. Our goal is really to give you a quick handle on what this platform is, what it does, and maybe why it matters if you're trying to, get up to speed in genomic variant analysis. And just so you know, we're pulling our info straight from the OakVar documentation itself.
Exactly, straight from the source. The docs lay out its main jobs pretty clearly. Things like annotating genomic variants, creating databases from them, querying those databases, reporting, and even visualization. That's basically our roadmap for today.
Okay, good. Let's start with that first one, annotation. The docs say it uses diverse annotation sources.
hmm
Why is that step one? Like, what does annotating actually tell you right off the bat when you're looking at raw genomic data?
Well, think about finding a typo in a huge book. Annotation is like asking experts, is this typo common? Does it change the sentence's meaning? In genomics, it's similar. Annotation looks up a genetic change in databases. ClinVar is a big one to see if it's known, if it's linked to any health issues. It gives immediate context.
Ah, okay. So it's not just here's a change, it's here's what we might already know about this change. Significance, basically. Then there's creating databases of these annotated variants. Why bother with a database? Why not just keep them in, I don't know, spreadsheets or something?
Yeah, good question. Imagine you have millions, maybe billions of these data points. The
Perfect.
database isn't just storage, it's structured storage. It lets you index everything, search really fast, cross-reference things. Trying to do that with separate files would be, well, incredibly slow and inefficient. It's like a library catalog versus just piles of books everywhere. You need the catalog to find anything specific.
Right, right. Scale demand structure makes total sense. And that structure enables the next point, querying variants using filter sets.
Hmm.
So once it's all organized, what kind of specific questions can researchers actually ask? What does this filtering let them do?
This is where it gets powerful for research.
Yeah.
You can get really specific. You could say, okay, show me only the variants in this gene that are rare, predicted to seriously affect the protein, and seen in people with condition X. Filter sets let you cut through all the background noise and zero in on the potentially important stuff.
Okay, so it's about precision searching, finding the needle in the haystack almost. The docs also mention diverse report formats. Why is flexibility in how you present the findings important? Who needs different formats?
Well, think about who uses this data. A geneticist doing deep research needs all the gory details, right? But maybe a clinician just needs a clear, concise summary for patient care. Or maybe you need output for a specific downstream analysis tool. Different needs, different formats. It makes the results more usable for everyone involved.
Tailoring the output, got it. And then there's the visual side, a graphical user interface, a GUI. How does actually seeing genomic data help? What do visuals add?
Oh, visuals can be huge. Sometimes patterns just jump out at you from a graph that you'd never spot in tables of numbers. You could visualize where variants cluster on a chromosome or compare frequencies between populations or zoom into a specific gene region. It makes complex data more intuitive to explore.
So it aids understanding and maybe even sparks new ideas just by looking at it differently. Now, OakVar works via command line, the CLI, and this GUI. Why offer both? Are there different users or tasks suited to each?
Definitely. The CLI is great for power users, people comfortable with scripting. You can automate complex workflows, run batch jobs, integrate it into bigger pipelines, very flexible. The GUI, on the other hand, lowers the barrier to entry. It's more visual, interactive, point and click. Good for exploration or for users who aren't command line wizards. Having both just makes it accessible to more people.
Right, caters to different workflows and skill levels. Okay, this next point sounds really interesting. The documentation calls OakVar an operating system for genomics apps. It says it lets people develop, run, and even distribute their own custom genomics tools. What does that mean practically? How does it work like an OS?
Yeah, that's a great analogy. Think of your computer's OS, Windows, Mac OS, whatever. It provides the basic functions, file management, the environment for other programs to run, right? OakVar aims to do something similar for genomics analysis. It provides the core engine for handling variant data, annotation, querying, and then researchers can build specialized apps or modules on top of that foundation to perform very specific analyses tailored to their own research. It's a platform for building new tools.
Ah, okay. So it's not just a tool itself, it's a platform for building other tools. That really clarifies its potential extensibility. And finally, they briefly mention linking genomic data to AI and machine learning models. Where could that lead? What's the potential there?
Well, that's looking towards the future, isn't it? If you have large, well-organized, annotated genomic data sets like OakVar helps create, You can then feed that data into AML algorithms.
Hmm.
The hope is to build much smarter predictive models, things like predicting disease risk more accurately, understanding why some people respond to drugs and others don't, discovering really complex gene interactions. It opens up a lot of possibilities.
Okay, so bringing it all together, OakVar seems like this quite versatile toolkit. It handles the whole workflow annotation, organizing data, searching it, visualizing it, and crucially, it acts as a base for developing new custom applications and potentially plugging into future AI developments.
That sums it up pretty well. It's designed to be a comprehensive environment to make genomic data analysis more streamlined and powerful.
Definitely sounds like a powerful system. Okay, that gives you a solid overview of OakVar. So here's something to think about. As platforms like this make complex genomic data easier to manage and analyze, how might that change the speed of discovery? What new questions might become answerable when these kinds of integrated tools are more widely available in biology and medicine? Something to consider. Join us next time for another deep dive.